Okay, hi everybody. For good and bad, this is the last session of the meeting, which is called Plant Physiology and Mechanisms. So I think that one thing we can all agree on is that plants are amazing organisms. And as a scientist, we are inspired by the large repertoire of sizes and shapes that they have, the incredible developmental plasticity, and the fascinating solution plants have evolved to survive an ever-changing environment. In this session, we aim to highlight the importance of understanding the fundamental mechanisms that enable plants to achieve these remarkable abilities at a scale ranging from the single cell to the performance of the entire plant. Such knowledge, we think, is critical to achieve the goal of this meeting, which is to improve crops at the face of climate change. So we heard really great examples of how this basic understanding is important, and in this session you will hear more about it from three speakers that mainly revolve around the physiological mechanisms. So the first speaker of today will be Menachem Meshalion. He is from the Hebrew University. He's an expert in the regulatory role of plant aquaporing in controlling the plant water balance. His research revolves around improving agronomic plant vigor and enhancing yield production. He's also interested in development technological advances in agriculture. And he will talk about discoveries regarding the impact of stomata dynamics in plant response to water regulation. The second speaker is Tamara Zulay-Shemer. She's from the Agriculture Research Organization that is located in the north part of Israel in the Veyar Center. She's an expert in photosynthesis and water balance, and her lab explores plant responses to stress, including temperature extremes, drought, and rising CO2 levels. Tamar is also a leader. She leads a fruits tree sciences unit at the Israeli almond breeding program. In this context, she will present today her research aiming to understand the physiology and the genetic basis for stem photosynthesis capabilities in wild almond, which is a trait that is not present in the cultivar ones. And last, we will have Lisa Ainsworth. Lisa is a research leader of the UCSDA ARS Global Change and Photosynthesis Research Unit, and a professor of plant biology at the University of Illinois Urbana-Champaign. She's an expert in photosynthesis responses. She applies integrative approaches from genetic to agronomy to address crop responses to global atmospheric change and potential solution to mitigate climate change through agriculture. She was awarded the National Academy Sciences Prize in Food and Agriculture Sciences, and she was elected a member of the National Academy Sciences recently. So today she will present her research efforts to identify the physiological determinants of yield change in response to elevated CO2 and ozone, as well as strategies to improve canopy architecture and photosynthesis. So I'm happy to invite the first speaker, Menachem Meshalion.
So, we all know this already, right? So drought is a major impact, and the big reason for this is transpiration. I mean, the only reason plants are so sensitive to drought is because they lose so much water. And the water loss via the stomata in molecular ratio is about 500 to 1 on average on croplands in Israel. That means the plant loses 500 molecules of water in order to gain 1 CO2, which basically make a very bad water use efficiency for crops. And when you think about it, this is not the status at wild type. In wild type, plants lose much less water, but breeding, which was meant to increase yield, also increased the leaf size and the number of stomata and the time the stomata is opening. So when you look at almost any crop, you see they transpire much more water. So the productivity is going up, but also the water is going up. And this is a very nice presentation. We also saw similar yesterday. This is the release of the bread. This is wheat. And you see the yield growing up. This is what we expect from breeding, right? But the same researcher took the same crops and then he tested stomatal conductance. And you can see that as more the plant produces, the more time the stomata is opening. This makes actually the plant, the crops, to take a lot of risk because they use the water a lot in order to get CO2, right, to open stomata. Now, stomata is a fantastic organism. In this slide, you see only the receptor of light wavelengths, right? From UV to far red in a single cell. So plant or guard cells sense the wavelength, the intensity, temperature, CO2, and of course, ABA. When ABA goes up, it's actually overrule all the other signals. So it doesn't matter how light, temperature. If ABA goes up, everything closes down. So we have multi-signals, and when I think of water balance, I think about this kind of a balance. It's a very multifactorial balance that it's very hard to maintain, right? So water balance in plants actually involve many factors of light, temperature, humidity, soil water content, light intensity and quality, and so on and so forth. So maintaining this at a balance means that the plant have to get by with whatever the environment gives them. And also, there are many strategies to respond to these lights, right? So many sensors, so many receptors. Probably there are also very different ways of responding, like in management ways. And this is exactly my research. We are trying to understand what is the management decisions of the stomata. Some stomata open very fast, close very fast, responds to light, don't respond to light, respond to CO2 faster, slower. And we believe that these tiny micromanagement differences is the reason that some plants produce a bit more and some plants produce a bit less. So what we want to see, so we also talked about this before, so breeding for abiotic stress is a very, very long process. And one of the reasons is this dynamics of the plant response and the dynamic environment. So our hypothesis was that if we can screen back plants that were already bred in the field and we already know their production, both under field conditions and in the lab, both under well-irrigated and drought conditions, maybe we can find some of the key physiological traits of this stomata, and maybe by understanding these key traits, we can also use this as traits for future selection. So this is exactly what we did. We took Professor Dani Zamir's tomato introgression lines. The nice thing that all of the plants are M82, but each cultivar has one piece of a genome from the Pinelli. So the plants are very similar, both genetically and morphology, but they have a very different behavior. Dani Zamir was generous enough to give us all the 10 years of field data, both from well-irrigated and drought. When I say drought, it's drought in Israel, so plant will die in drought of Israel. So these are irrigated, but controlled irrigated. So we talk about mild to moderate drought, not a catastrophic drought. So we took all of this data, and we screen all the plants, and we look for plants that behave better than the M82. And we look for several behaviors, like you can see here. This plant, this 5-1, actually has always better both under drought and control from the M82, so we call it idiotype. And I want you to focus now only on this 5-2, the green one, and the red one, because we have many, so it will be simpler to follow. I will show you all the results, but it will be simpler to follow on these two ends. And we took another one, which is 8-1, which was always lower in productivity and much more susceptible to the drought. Now, And we make sure we had at least 4 to 5 years of experimental from the field, so we know we can maybe a bit trust the data. How to screen the plant? So there are many ways. We all know these cameras. I don't think these are very good for physiology. Today I'm going to talk about a screening that's called functional phenotyping. I will explain more. But I decided to bring some paper we released lately as editorial in the Journal of Experimental Botany, where we decided to set up some guidelines of how to do controlled drought experiments in pots. So why me and 4 of my colleagues decided to write these guidelines? Because we get many papers on drought, a lot of them. Most of them have some critical issues of just in the experimental design. I'm not going to go too much through it because I want to show the result of the screening we did, but some of the principles we follow. So we call it the 4 Ds. You need to define your stress, try not to go to very general, like we want to screen for drought resistant, blah, 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 but try to be very quantitative when you do it. When you develop your hypothesis, try to use the right vocabulary. I want to give you some example. When you have the stress, so usually stress started and then the plant could die, so it's susceptible to the drought, it's not sensitive to the drought, because if the plant were sensitive, you would probably sense it ahead and respond to it by either escape or other mechanisms. Now, if you respond and survive, so this is a survival plant that actually passed the stress, maybe rain come, maybe we irrigated. And then there is the resilience or the recovery. So resilience is the recovery from the stress. Now, all of these, or even this plant could be considered as resistant if we talk about ecological resistance, because the plant did not die. But from the agronomical point of view, this plant actually has a low tolerance and a low resilience, because it just survived. This is not agronomical trait at all. I'm not going to go to all the details, but also resistant could be only plant that look exactly like control in the yield amount. So if we talk about agronomical point, we usually talk about crop and not just, many times you see in pictures that people show you, okay, this plant did not die. So from agronomical point of view, that doesn't mean a lot. So how do you do this screen? I will talk later on the phenotype, but now I want to talk to you about some big artifact or the major artifact in pot drought experiments. So one of the biggest artifacts is comparing the stress. When you want to do stress for many plants, you want to make sure they're suffering from the same stress. So let's look on a simple experiment. We have a pot, let's say we have 500 ml of water, we irrigate it, and then we let the plant transpire. Let's say the plant transpire 140 ml. So the next day you have only 360 ml, but it grows a bit, so it transpires a bit more the day after, 150 ml. So the next day this plant has only 210 and then 90, and then we see some wilting because the plant doesn't have water to transpire. Now let's say that in other point of the table, when there are only 1 degree more, this plant transpires only 110. We don't know it unless we measure it. That means that this plant will have much more water than the other plant, let's say at day 3 or day 4. Now when we compare the plants based on the day, we actually compare them to different stresses. So taking day in account, especially if you want to collect the RNA or other metabolites and comparing the plants based on the day without knowing the transpiration, this is a big, I would say, biased. So this is how the introgression lines that we work with looks like if you just close the tab here. This is the soil water content. You see some plants almost don't lose the water content and some lose it very fast just because of transpiration differences. Okay? And it could be from the environment or from the genetics. So a small plant or a small transparent plant could be here, and the higher transparent could be here. So of course that after a few days, if you just collect the data here, you are not going to get the right comparison. How do we control it? We control it via special controllers that actually irrigate the plants. We use the plant-based system and we irrigate the plants based on their own transpiration. So we have a feedback system that brings back water to each pot separately based on their own plant transpiration. This system also has covers both from the bottom, so nothing leaks either from the bottom or from the top. Each plant has its own controller, soil sensor, VPD and light sensor, so we know, and irrigation system. So each plant is being irrigated separately based on its own transpiration. Each plant sitting on a lysimeter, so we know the water loss rate for each plant separately. And in the end, this system enables us to reduce the noise coming from a lot of things in the greenhouse and get pretty much clear signal. Because each plant has also its own controller, we can build a truly randomized experiment because we don't need to split the pipes. Each plant getting the irrigation based on its own transpiration. So each plant, actually, we can control the irrigation. We measure several physiological impact, and all the plants are exposed to the same environment. So we make sure they have the same environment and the same soil water content. So they are actually really exposed to the same potential all the time. And we measure all the plants simultaneously and continuously via the drought and the recovery phases, where all the plants sees all ambient conditions. So this is what we call the functional phenotyping. We actually measure continuously the plant biomass gain, transpiration, water use efficiency, stomatal conductance. I'm not going to talk today about root influx and the drought point. I will show you this result soon. And it's absolute measurement, so it's not like a porometer or Likert that you measure a piece of a leaf, you measure the whole plant. So let's go back to our screening. We want to compare the plants. So this is 8.1, you remember the poor looking or the poor behavior plants. So this is day by day, this is the daily transpiration, how much water in gram the plant transpires per day. And you can actually see the plant growing. We don't see biomass here, but we see functional trait. When you look on the idiotype, you see it transpire much more, and I want to remind you, this plant produces more even under drought. So this is a big surprise. How come a plant that loses more water in the end produce more? I'm going to, and this is the M82, this is like, let's say, the mother plant somewhere in the middle. And these are all the other plants. I don't have time to go now to details, but we can later, if you want, can discuss the difference between all the lines. And this is the soil water content. So all of them under the soil water capacity all the time. We make sure the water are available at this part of the experiment, no drought at all. Then we expose the plant to drought. Look how nice the drought look. Because we use the feedback irrigation, so it doesn't matter if this plant you see it's lose twice than this, but the soil water content go in the same rate because the feedback irrigation. And then we have a recovery phase, so the plant allowed to recover, and we measure how much time it takes the plant to go back to its previous transpiration. You see it's few days, but you see a big difference between the line. Personally, I believe that this recovery is one of the most important traits to define drought response. Because if the plants activate many mechanisms here, it's all going to benefit here. During the drought, you hardly see anything. There is one thing you see, and I will show you soon. So we compare the drought and the recovery. Now, what is drought? Okay, what is stress? This is, again, it's a physiological point. So we define stress not based on the time, but based on the soil water content, the start limit transpiration. So on the y-axis you have transpiration, and on the x-axis you have the actual soil water content. This point here is where the water content in the soil become limiting factor for transpiration. To me, this is the physiological point. No other trait really show you this limitation like the water content that start limited transpiration. And of course you compare all the plants on the same trait and look for the different trait. But when you want to compare them under drought, it's the best to first to be after the tetacrit, but second under the same stress. So we choose a phase that all of the plants were under similar stress and below the tetacrit. So we know they are stressed, the closed stomata. Okay, and we try to make them as in similar stress as possible. Again, it's very important when you compare apples to apples. So what do you get when you measure this? Now I'm going to the resolution of minutes from day to minutes, so you can see if we take the transpiration rate, this is transpiration rate, and this is stomatal conductance. I'm not going to get to details now, but this is a very critical point that actually calculate the stomatal opening, but not based on looking, but based on the calculation. Stomatal conductance, like your porometer gives you. So this is the 8.1. It's look nice. But when you look on the 5.1, you see very interesting response. You see that in the morning, when the VPD is low and the light is sufficient, they fully open the stomata, although I want to remind you, they are now under 50% route. So this plant has dynamic water use efficiency where they open stomata at a certain time of the day, much wider than the other plants, and then they close it down. This transpiration shows the actual water loss. So this behavior allows them to lose less water, although they open stomata wider, but only like 1 hour a day. Is it enough? Probably yes, because in the end, as I show you, they produce more. So this 1 hour a day probably accumulates through the season and allows them to, in the end, produce more. So they're under the same stress, but behave differently. We did it to all our lines, and we wonder why would we have this different stomatal conductance behavior. So we went and we looked at the stomata. We just used this resin that you put on the leaf. We did it 8 times per each line during the day in a greenhouse because we want to reflect as close as possible to the measurement, to a real world measurement. And we did it to the abaxial and adaxial leaf, so both bottom and top. And you see very interestingly that the total density, the gray line, was hardly different between the lines. But, so if you take both top and bottom, no significant difference. But when you compare the top to the bottom, the 5.2 has much more stomata on the bottom of the leaf than on the top, while the 8.1 has a similar ratio between the top and the bottom. When you look on the stomatal aperture, okay, we really measure it in the microscope, you see that the 8.1 fully open stomata at noon, which is not a smart thing to do in Israel. While the 5.2 only outdo it in the morning, as I showed you before. So what the system shows is really reflect the real stomatal aperture. And I want to remind you, it has much more stomata on the bottom of the leaf. So theoretically, the same line, M82 just cross, and you see totally different stomatal pattern and stomatal behavior in a different time of the day. So, the difference is in the small details of the behavior. Of course, we did it to all the lines, and we did another thing. We look for different expression for both speech. It's a gene that regulates stomatal development, and the ZEP, which is actually related to ABA production. I will run a bit faster here. But what we found is very interesting. There was a nice correlation between the speech expression and the number of stomata at the abaxial part. And the opposite for the ABA. So the 8.1 has much more ABA probably. We didn't check the ABA, only the gene. And the 5.2 was the lowest. So it seems like ABA is responsible to the dynamic behavior, and speech 1 is for the number of the stomata in both sides. We didn't prove it, it's just a correlation. We also, at the end of the experiment, we cut the plants, we dry them, and we check the biomass. So you can see clearly also on the lysimeter, this 5.2 and has much better biomass, just dry biomass, after 4 weeks of experiment, and the water use efficiency. Water use efficiency, it's agronomic water use efficiency, which means we take the dry mass of a plant, And because we know the total transpiration of this plant throughout the experiment, we divide the total ml, it's in the gram ratio, of the plant with the total dry matter, and then we get agronomic water use efficiency. It's actually agronomic transpiration use efficiency because we cover the soil, so it's just the transpiration. So to sum up, what are the traits, the physiological traits that we think that are important for a better stress response? You see, I don't like the word resistant or tolerance. It's just a better stress response. So first, fast growth, so when the water are well, if the plant grows fast, it's just better, more leaf, more photosynthesis. Then, very high water use efficiency. So this fast growth is actually translate well to also nice absorption of CO2. Fast stomatal closure, so when the drought stress, they respond and close stomata. Much higher concentration of stomata, or stomatal density higher at the lower part of the leaf than the upper part of the leaf. This golden hour, we call it, so using the daily VPD and light in a way that will be much more efficient for the plant, so open the stomata when the VPD is low. So we call it dynamic water use efficiency. And recovery. As I told you, recovery was fantastic because this plant transpired almost twice in this. And even though you can think that the stress was more severe to it because it's actually a bigger plant, bigger leaf, but then it recovered faster, and this is like 4 or 5 days. So this resilience or recovery, I think, is one of the interesting traits. Okay, so I just want to thank my collaborators that helped me writing the 4Ds paper. Sanbon Goza was the PhD student that make actually most of the experiment here. Professor Dani Zamir actually gave me all the data from the field. Without it, we cannot do it. Bogala, Ravi, and Ramon actually was master student that helped in the other part. Thank you.
So thank you. First, I would like to say that it's a really great honor to be here today, and I would like to thank the committee and the organizer for giving me this opportunity. And we are all here because climate change calls for new strategies to mitigate its challenges. And today I would like to share with you one of the stories, a very interesting story, I really like it, that illustrate the value of using wild species, traits that come in from wild species into breeding. And I will share with you a part of the almond breeding. It's a part of the almond breeding program that focus on adapting commercial almond cultivars to climate change through the integration of stem photosynthesis trait from the wild almond Prunus arabica. So almond is an important agriculture crop in the world and is known for its health values and nutritional values. It is rich in monosaturated fats, in vitamin E, packed with fiber and antioxidants. It's good for our heart health and skin health, and for blood and sugar control. We are all aware of the climate negative effects on plant-based food production. And based on the FAO, the Food and Agriculture Organization, with the increase in the world population, which is estimated to reach 11 billion by the year of 2100, the food insecurity is increasing, and now it affects around 30% of world population. As deciduous fruit trees, almond trees goes into dormancy during the early winter, And during the winter, there are cold hours, which are known as chilling requirements that are very important for bud development and flowering. And during the spring, when the temperature starts to rise, they induce blooming and the regrowth of the trees. So one of the main effects of global warming is the warm winters. And with warm winters, there is a reduction in the number of chilling hours that affects the bud development and flowering. And in addition, with the warm winters, there is an increase in metabolism and the energy demand of the trees. So together, these environmental challenges, they are negatively affecting the flowering, productivity, and the fruit yield of almond trees and other deciduous fruit trees. So these conditions emphasize the vulnerability of almond orchards to climate change challenges and the need for adaptive strategies to mitigate these challenges. So Zion already shared his work and the importance of using wild species, bringing different traits from the wild. During the development of commercial cultivar, we are actually losing a lot of traits that do exist in wild species, with the climate change challenges, identifying these different traits for higher resilience, increasing in productivity and yield are very important and are in great demand. So in recent years, a rare almond species, the Prunus arabica, was added to the Neve Yaar almond collection, which is currently holding around 150 different types of almonds from different parts all over the world. And this Prunus arabica is a distinctive almond which was found around the Fertile Crest Mountains, and as such it is grown under very harsh environmental conditions. On one side it's grown around arid and semi-arid habitats under hot and dry conditions, and on the other it's been found in high altitudes and is growing under severe cold winters. So one of its morphological traits attracted our attention, and unlike the commercial and other almond types, its stems are green and they remain green and unbarked all year round. And we then decided to compare between the wild almond Prunus arabica and the commercial cultivar Prunus dulcis. We chose the Fachem, which is a very common commercial cultivar in Israel. And when we compare the surface area of the above ground tree, we found that the Prunus dulcis have 93.6% green leaves, and the Prunus arabica it's only 32%. But when we compare the area of the green stems, we found that in the Prunus dulcis it's only 0.1%, and in the Prunus arabica it is 60%. So, interestingly, there are different plant species, and in particular there's shrubs which use these green stems for photosynthesis. And they do so in particular under stress environmental conditions like hot and drought conditions. And then these stems plays as a major carbon source tissue, especially when the plants don't have any leaves. So these results led us to the following hypothesis that Prunus arabica, almond, assimilates CO2 photosynthetically via its stems, which give him another tissue for carbon assimilation and the energetic state, and keep its energetic state. So to test our hypothesis, we monitored stem gas exchange in both almond species, the Prunus arabica and the Prunus dulcis, the commercial cultivar, and we found that the net CO2 assimilation from the stems, this is not leaves, it's stems, it's significantly high and impressive all year long, also during the winter when the tree has no leaves. These levels are actually 80% of the net CO2 assimilation of the leaves of this Prunus arabica, which is very high. So to reveal which component contribute to this distinctive photosynthetical potential, the wild almond Prunus arabica stems, we conducted different physiological and histological analysis to compare between these two almond species. As you heard here a lot, for photosynthesis, the plant need to use stomata for gas exchange and for assimilating the CO2 from the atmosphere. And therefore, we decided to check whether the stems have stomates. We analyzed the Prunus arabica and the Prunus dulcis, the commercial one, the first ear stems and the second ear stems of development, and we found that a significant difference. The Prunus arabica stems has threefold more stomates on the first ear stems and also on the second, but the Prunus dulcis stems in the first ear, they have very few stomata, but on the second ear, it was covered under a very thick barked layer. So they're actually disappearing. So next we wanted to explore whether these stomata are functional and respond to different environmental stimuli. And we used gas exchange measurement and measured their response to CO2, to light, and to temperature. And as you can see, like the stomates in leaf, they are fully functional. High CO2-induced stomatal closure, low CO2-induced stomatal opening. The light to dark transition, you can see that the stomata are closing following dark, the exposure to dark. And temperature, high temperature induced the stomata on the stems of the Prunus arabica to close. So next, as I mentioned, the Prunus arabica stems, they are green and unbarked, and we therefore wanted to check this further. We made cross-sections of the stems, and we visualized them under fluorescent microscopy. And you can see here that the Prunus dulcis, the commercial one, they have suberized autofluorescence cells of the cork phylum, which is responsible for the bark formation, and we didn't observe this in the Prunus arabica stems under this developmental stage of stems. When we did some histological sections and used safranin O and fast green to stain them, we found some significant differences between these two species. You can see here the Prunus arabica stems. They are rich in stomata. You can see here the stomate, which is open up to a very thick parenchyma layer, and this parenchyma layer is called pellucid-like parenchyma because it really resembles to this pellucid mesophyll, which is responsible for the photosynthesis in leaves, and they are rich in chloroplasts. On the other hand, when we look at the stems of the Prunus dulcis, the parenchyma is round, the cells are round shape, they are quite dense. You can see here the bark layer and the periderm, which was not observed on the stems of the Prunus arabica. And in addition, something really interesting, which we are following up now, while the Prunus dulcis have one main vessels, phloem and xylem. Here, except for the main one, you can see here 5 additional cortical bundles, and we believe that they are also responsible for some of these unique stem photosynthesis traits. So we also found some differences on the chlorophyll levels, which was significantly different, also by the levels and also by the distribution. In the Prunus arabica, it is located mainly to this pellucid-like mesophyll, pellucid-like parenchyma layer. And on the Prunus dulcis, there is chlorophyll, but it's under a thick bark layer, and it is distributed along the different tissue layer, from the parenchyma to the phloem and xylem, and it gets until it gets to the pit. So, based on this result, we hypothesized that this unique stem photosynthesis trait may be beneficial for deciduous fruit trees, which function as a carbon gain tissue, in particular during the dormancy when the tree has no leaves. Furthermore, this tissue can provide with additional energy that is crucial during warm winters when the plants have higher metabolism and energy loss. So, can we develop a winter photosynthesizing almond cultivar to enhance energy reserves and yield? So, to uncover the genetic and mechanism behind this stem photosynthetic trait from Prunus arabica, we generated a crossbreeding F1 population by crossing the wild almond Prunus arabica together with the commercial almond cultivar Prunus dulcis. This is El Fachem, and here you can see the progenies. We got approximately 100 F1 progenies, and here you can see Carmel, our orchard manager, between them. And next, we analyzed using gas exchange the stem net CO2 assimilation for all these progenies in the F1 population. You can see here also the parental lines, the Prunus dulcis and the Prunus arabica, and you can see the nice distribution. Pretty low, pretty high, the parental lines. And next we sequenced the genomes of both the parents, the Prunus arabica and Prunus dulcis, and we use approximately 5,000 informative SNPs markers for QTL analysis and for GWAS, and we found 2 QTLs, one on chromosome 7, which was a major QTL, and the loss score was very high, around 20, and and this one was able to explain approximately 67% of the stem photosynthesis phenotype, and another minor QTL on chromosome 1 with a lower load score of 4, which was able to explain 90% of the stem photosynthesis phenotype. Next, we generated a list of 64 genes that are located around these two QTLs, which are non-synonymous in the coding region. And from them, we found two very interesting candidate genes, the HHSD type acyltransferase family protein, and another one, the MIB family transcription factor. Both of them are known to be involved in bark formation and epidermal cells regulation. So we are following on top of them. So the most important thing is to know if we can use this for breeding. So is the stem photosynthesis capability trait is beneficial for commercial almond breeding? And for that, we use our F1 population, and we measure them for a trunk diameter, for a blooming time, and for yield. And as you can see here, the trunk diameter was found to be significantly higher in those progenies that had the stem photosynthesis capability trait. In addition, we found an earlier blooming time for the one that had this trait. And last, which is the most important, the yield was higher. And another point, this was measured for 3 years in a row. So this gave us a really good feeling that we are on the right track. All this data was published in different publications. In addition, the use of the stem photosynthesis to increase yield is protected under a patent in both Spain and the US. And with the progress of the study, we also are now looking at advanced populations, and it seems to be a little bit more complicated. So this stem photosynthesis trait is not a one gene trait, and now we are following on first, the genetic inheritance and association between the different elements of the stem photosynthesis. And in addition, since this one is a part of the almond breeding program, we are incorporating this trait into different commercial cultivars, and we check them for their resilience and yield under this, the climate change challenges. So this work actually set the stage for future breeding for cultivars that will be better resilience. Is it fine resilience, Menachem? With climate change challenges. And with that, I would like to thank you for your attention, and of course to all the people that did all this work, to Dr. Doron Holland, which was in charge of the genetic aspects of this project, to all the people, Kamel Khatib, our orchard manager, to Rotem Beja, our geneticist, Or Shapira, physiologist, Tali Tronin, our lab technician, to Hillel Bruckenthal, which is a former PhD student, which did most of the work, Zivati, a former postdoc. We have now 2 students that are continuing this further, Uri Eisenbach and Dan Zeira. Irit Bar Yaakov is a volunteer. All our collaborators in Bedagan, Dan Charuvi, Vivek Tuari, Edward Basarov, Chani Tzemach, and Shira Gan, and of course all the funding agents that without them it could not happen. So thank you.
Thanks, everybody. Last talk, we're almost done. So thanks very much to the organizers for the invitation. This has been really an exceptional meeting. And so I'm going to talk about optimizing canopy photosynthesis for atmospheric change. And so, of course, you all know that the atmosphere and the climate in which crops are growing is changing and changing really rapidly. And NASA calls this figure the relentless rise of carbon dioxide. And so CO2 concentrations are higher than they've been for over 1 million years. And if we think about how long agriculture's been around, it's sort of this little blip down here. So our crops, we first domesticated them when CO2 concentrations were really much, much lower than they are today. And of course, it's the increase in CO2 concentrations that has caused much of the temperature rise that we've been talking about. And that has then impacted the hydrological cycle, which we've also been talking about. But the increase in CO2 is global. It's happening everywhere on the planet. And CO2 isn't the only atmospheric component that's changed. Ozone is another pollutant in the air. Most of the ozone, of course, is in the stratosphere, where it protects us from UV radiation, and that's a good thing for those of us that have DNA and proteins. Not all of us anymore because of AI, of course. So ozone in the troposphere, where we live and breathe, is a pollutant, and it's a component of photochemical smog. So we don't have the same kind of long-term records from ice cores of ozone, but we do have some estimates of pre-industrial concentrations of ozone. And the best ones are probably from Paris, and suggesting that background concentrations were close to about 10 or 11, between 10 and 15 parts per billion. And so today concentrations are 4 to 5 times higher than that. And while in some parts of the world concentrations have stabilized, in other parts of the world background ozone concentrations are continuing to increase. And so both of these pollutants impact crops and crop productivity. And I think as we look forward into the future, what's important to remember is that even if we reach peak CO2 emissions today, which we probably won't, concentrations will continue to increase at least through the middle of this century and likely through the end of this century. And so it really will take very stringent policies to reduce CO2 emissions to bring this down, to bring CO2 concentrations down. And similarly, ozone concentrations, the tropospheric ozone burden, is likely to increase as we move forward. So we've heard about the importance of stomata today for pathogens, and also my water arrow should be a little bigger, sorry. But stomata are also critical because that's obviously the diffusion pathway for carbon dioxide. And so when we increase carbon dioxide in the atmosphere, what that can do in C3 crops is stimulate photosynthesis and outcompete the photorespiration reactions, and that increases yields. The same diffusion pathway exists for ozone, and so ozone also diffuses into leaves, but unlike CO2, it has a negative impact, and so it accelerates the process of senescence, meaning that the cells are aging more quickly. That reduces photosynthesis and has a negative impact on yields. Both the CO2 and the ozone cycles have greatly intensified over the past 100 years. And today, terrestrial ecosystems take up about 1/3 of the CO2 that we're putting into the atmosphere. So that's really a tremendous filter to our emissions. And they also take up about 1/5 of tropospheric ozone concentrations. And so the capacity of our ecosystems to continue to filter these pollutants is really important. It has critical consequences for future climate and also for the provisioning of ecosystem goods and services, including crop yields. And so what I'm going to talk about today is why we think photosynthesis is the key trait for maximizing crop responses to these changes in the atmosphere. And there are a few key points that I want to make. First, that photosynthetic rates are both supported and constrained by environmental conditions, and we've heard about that today. Photosynthesis is well described mathematically and biochemically, and so targets can be identified through modeling and then tested through experimentation. And in the context of rising ozone, the response of photosynthesis to ozone pollution is correlated with the seed yield response. And so stabilizing photosynthesis under this pollutant is a key target for improving yields. And then when we think about rising CO2, we have to think about the history of breeding as well. And so breeding has increased the harvest index, or the sink size, in most of our major crops. And so that means that there's probably capacity now to increase photosynthesis in order to improve yields. So I was really inspired, I think everybody was, Michelle, by your talk about being in the field. And really, there's no place I'd rather be than in the field either. And we have studied the response of crops to rising CO2 and ozone pollution in the field as much as we can for the past 20 years. And so this is an aerial image of the soy face experiment that's in Champaign, Illinois, where each of these octagons that you see is a location where we can release either CO2 or air that's enriched with ozone into a plot and then grow crops under different atmospheric conditions. And so if we zoom in on one of these octagons, what you will see is an array of pipes, and these are perforated pipes that have small holes all throughout the length of the pipe. They're connected to a manifold here that has valves that open and close and control which side of the ring we're emitting CO2. That's controlled because we're taking measurements every 4 seconds of the CO2 or ozone concentration in the center of the ring. That information is relayed to a computer that controls then this manifold. And so we're also measuring the wind speed and wind direction. So if the wind is coming from the direction of the yellow arrow, then these pipes will be open, and then we will release gas into the wind, and the eddies of wind basically carry the gas across the plot. And again, every 4 seconds we're making measurements, and so if the wind speed or the wind direction changes, then we can change the system. And in this way, we can grow crops under higher CO2 or higher ozone concentrations for the whole season. And so for our ozone concentrations, we've both monitored the background ozone and then added a proportion on top of that. For screening experiments, we've also had a target concentration in more recent years of about 100 parts per billion. And while our background concentrations are shown here in blue, and they're about 40 parts per billion in Champaign, and 100 is more than a twofold increase, that's a concentration that regularly occurs in polluted parts of the world. And so you can see ozone concentrations in China here that well exceed 100 parts per billion. For our CO2 experiments, we started with a concentration of about 550 parts per million, which is what's anticipated for about the middle of this century. We've increased that to about 600 parts per million as our background concentrations have increased. Okay, so I want to first talk about our experiments with ozone and why we think improving photosynthesis can also improve yields in elevated ozone. And so one of the key hallmark responses of crops to rising ozone concentrations is that they accelerate the process of senescence. So what this graph is showing you is the rate of photosynthesis of a number of different maize hybrids versus the day after anthesis. And so a couple of things that I'll point out is that when you grow plants under ambient conditions, and so those are the open symbols here, rates of photosynthesis are high, and then as the leaves age, they naturally lose the photosynthetic capacity. And so that's this process of senescence. In elevated ozone, which are the filled-in symbols, what you see is the rate of photosynthesis early on in a young leaf is often about the same as in a leaf grown under ambient ozone concentrations, but they start to senesce sooner and they senesce more rapidly. You can integrate under these curves to estimate the amount of carbon that is lost under elevated ozone, and so it can range from about 13% in some lines to almost 50% in other lines. And this is a response that we see in corn. That's what I'm showing here. We see it in wheat and in rice and in soybean as well. And so we've also looked at the metabolite profile in these aging leaves. And so early on, if we look at the metabolite profiles just using metabolomics, we don't see any difference in ambient and elevated ozone. But as the leaves are aging, then these differences appear. And one of the key responses that we've seen using metabolomics is that that ROS scavengers like alpha tocopherols and also membrane stabilizers like campestrol and stigmasterol increase in response to elevated ozone in these aging leaves. And these are also components that can protect the photosynthetic apparatus. Importantly, the percent change in photosynthesis that we measure is correlated to the percent change in kernel mass that we measure as well. And so again, maintaining photosynthesis is a good way to maintain yields. And so, as I mentioned, I showed you data from corn. We've also done experiments with soybean, and we've done experiments where we added other stresses as well. And so this is recent work where we grew soybeans under elevated ozone, but also used these awnings to capture rainfall. And so plants were grown with lower soil moisture as well as ozone. And we did this experiment for 3 years. And what we found was, regardless of whether they were grown with reduced soil moisture, and we reduced the soil moisture by about 40%, elevated ozone, which is shown in the red symbols here, caused a decrease in yield. And so there's some hypothesis that under a reduced soil moisture or a drought condition, because stomata close, ozone wouldn't have a negative effect. But that's not what we see. Even in the presence of drought stress, ozone is still reducing yields. So we think that maintaining photosynthetic capacity is a key target for improving crop responses to ozone. So just to summarize this part, accelerating senescence is a hallmark response of crops to rising ozone pollution. We think that maintaining photosynthesis is a key target, and that a component of that is increasing these ROS scavenger molecules in membrane stabilizers. So I'll move on to the last part, and that's that breeding has increased the harvest index, providing a larger sink for photosynthate that's produced in elevated ozone. And so I'll just start by reminding you that yield can be broken down, and this is from the Monteith equation, as a series of efficiencies. And so the yield potential in any location is a function of the amount of light that that location receives, and the amount of light that can be used by photosynthesis, multiplied by the efficiency by which leaves or the solar panels intercept that light, or the interception efficiency, multiplied by how efficiently plants convert light energy into carbohydrates, and that's a function of photosynthesis and respiration, multiplied by the partitioning efficiency or the harvest index. And so these three efficiencies combine to determine the yield potential in any given location on the planet. And so we've seen plots of yield gain over the last couple of days, and we've seen it for corn, and we saw it for wheat, and so now I'm showing you a plot of yield gain for soybean. And so this is, again, these are experiments where we've grown different varieties of soybean, and YOR is the year of release. So these are soybeans that were released from the 1920s to 2013, and they were grown in a common garden environment. So same management, same density, and this is the rate of gain that you see. So yields have more than doubled with breeding efforts, and that's a testament to the soybean breeders, obviously. So when we look at the physiological basis, or which components of the yield equation have contributed to this, there has been a slight increase in interception efficiency, so they're more efficiently capturing radiation. There's been a very small increase in the conversion efficiency, and there has been a big increase in the harvest index. And so that has contributed the most to soybean yield gains. And so, again, this is one of the rationale for why we think photosynthesis is a good target, because now we have a big sink for that extra photosynthate. Over the past 20 years, we've looked at over 200 different soybean varieties in the face rings that I showed you before, and tested how they respond. And we also know that the lines that have the greater proportionate yield increase tend to have a greater yield potential. We've looked in particular at a couple of lines, LODA and HS93, which LODA has a greater than expected yield increase in elevated CO2, and HS93 4118 has a less than expected yield increase. And so we looked again at the physiological determinants of their seed yield and elevated CO2. And this is what we found. We found that LOTA, on average, has a lower interception efficiency and a lower leaf area index than HS93, which is the line that doesn't respond, but its leaf area index and its interception efficiency increases at elevated CO2, in part contributing to the increased yield. LOTA also has a slightly higher conversion efficiency, and it has a higher harvest index, and it maintains that higher harvest index at elevated CO2. So all of these traits contribute to greater yields and elevated CO2. I think key here is that it has a lower interception efficiency and lower leaf area index. And so that leads to the hypothesis that lines that have a lower leaf area index can have a greater response to elevated CO2. And so soybean has a lot of natural variation in leaf shape and leaf size, and so we can capitalize on that to alter the leaf area index. And so this is exactly what we've done. This variation in leaf shape is very well known in soybean. We screened some of the germplasm collection that's present in the USDA collection that's in Champaign, Illinois for leaf shape, and found that the individual leaf area is correlated with the leaf ratio, and the ratio is just the length of the leaf relative to the width. And so this has a leaf ratio of 3.15, and this nearly round leaf has a ratio close to 1. And so lines with a lower leaf length to width ratio have high area, ones with a high leaf to length ratio have a low leaf area. And so the work that we've done over the past few years has been to develop a population of lines that are in a high-yielding background, but have a difference in leaf shape. And so what we did was we crossed a line that Brian Deers, who's a breeder at Illinois, has developed that has very high yield with these plant introduction lines that have the narrow leaf phenotype. And so I'll just say that the narrow leaf phenotype is also associated with adaptation to higher altitude, or not higher altitude, but higher latitude, so short day length. And so these lines that have a narrow leaf shape also don't yield very well in the location where we live in Champaign. And so that was the rationale for back crossing this trait into this line. We also know that these lines with the narrow leaf shape have lower leaf area, but key to this is they had lower leaf area index at the sort of reproductive stage, and that was a trait that we really wanted to select for. These narrow lines also are thicker. They have a thicker palisade, and so they have greater photosynthetic capacity per unit leaf area as well. And we haven't done the biomass. We don't know whether there's a difference in mass yet, but that's what we're studying now. So what we did was we back-crossed these lines for many generations, and we then put out in the field last summer BC3 lines and some BC4 lines, and we grew them under narrow row spacing or 15-inch row spacing, and then wider row spacing or 30-inch row spacing, and checked their leaf area index. And under both conditions, both a narrow and a wide row spacing, changing the leaf shape decreased the leaf area index by over 1 unit. And so we know that changes the light environment within the canopy. We measured that as well. When we looked at the yield of these lines, we also know that the yield was not impacted. And so some of our highest yielding lines did have the narrow leaf phenotype on average, because we tested over 100 narrow leaf lines and 100 broadleaf lines. On average, there's no statistical difference between them in yield, but some of our highest yielding lines had narrow leaves. So we think that this is a resource that we can now use. We have the same genetic background. We have differences in leaf shape to test other approaches to improve photosynthesis and to test them for their CO2 response. So we think photosynthesis is a key trait for maximizing crop responses to CO2. As I mentioned, rates are supported and constrained by environmental conditions. The process is well described mathematically, and we know that the CO2 response to photosynthesis, or the ozone response to photosynthesis, is correlated with seed yield. And we now have a system where we can test with a large sink the impact of greater canopy photosynthesis. So with that, I'd like to acknowledge everybody who has contributed to this over the years and my current lab group, and I'm happy to take any questions.